Today we're going to have a talk, which is the combined presentation. And we're going to present Curing Sickle Cell Disease, the City of Hope approaches. And we do have evaluations form today. Please fill the blue ones. And courtesy announcement, please put everything on vibrate. And please do not put it on the table because the table vibrates. Disclosure information, everything is, our disclosures in the back of the blue form. And the next ground rounds are going to be next week on September 11, and it's going to be Schwartz Center rounds in memoriam, someone here I will never forget. And we have 3 speakers today, myself, it's hard to introduce, then Dr. Cardoso and Dr. Rosenthal. And just because maybe Dr. Cardoso, it's not, we don't see him very often in our clinical presentations, and I'd like to introduce him in a little more details. He is the research professor, Center for Gene Therapy, and he joined the City of Hope in 2016. And he was educated in Portugal, where he received his medical degree. He earned his PhD from the University of Paris after completing doctoral research work in France, Portugal, and also here in the US at the Harvard Dana-Farber Center Institute. He spent a decade at Harvard Medical School and Dana-Farber Center Institute as the instructor of medicine, then taught medicine and molecular genetics at Indiana University School of Medicine. And he focuses on genetic mechanisms of leukemia, especially acute lymphoblastic leukemia in children. And right now also he has a lot of interest in the sickle cell disease, as he is going to present today. And Dr. Rosenthal, I think Dr. Rosenthal does not need the introduction. He came to City of Hope in 1996 when he really built up our pediatric bone marrow transplant program. And for 2 years, 2009 to 2011, he was the acting chair of pediatrics. And in 2011, he became the chair of pediatric and director of our pediatric hematology oncology program. And this is what he's building right now. And additionally, need to back a little bit, he received the CME grant for 5.7 million to develop the cure for sickle cell disease. And I joined City of Hope in 2000 and became the director of the bone marrow transplant about 10 years ago and work as a team. Thank you. And I think Dr. Rosenthal is happy.
Good afternoon, everyone. Thank you for coming. And what we want to accomplish in this meeting is to give you a sense of what is sickle cell disease. Most of you know from medical school or nursing school. What is it that we are doing, and why are we doing it here at the City of Hope? I have nothing to disclose on the part of the work that I will present. Here is sickle cell disease. You can see on the right corner here, sickling, and what the cause of disease is a simple exchange in the 6 codon of the beta chain from sorry about that. The 6 from GHG to GTG and glutamine acid to valine substitution. And what that produces is a state of poor affinity to oxygen, and therefore the loss of the shape of the red cell. So if you have the red cell usually has excess of membrane to volume, it can move through very tiny tunnels. If the red cell becomes rigid, it loses its ability to transform and to move through very narrow spaces. And this is what causes most of the complication related to sickle cell disease. What are the long-term organ damages? Classically, neurologic. 11% of the patient will have stroke before the age of 18. A third will develop silent stroke by age 15. We just saw a patient on Friday. She's a college student, and a recent MRI showed that she had a silent stroke. Untreated, she's being treated by exchange transfusion. Untreated, she will end up with heavy debilitation. Pulmonary issues, acute chest syndrome, vaso-occlusion of the blood flow in very small vessels in the lung, pulmonary hypertension, 40% of the patient by age 40, renal insufficiency, infections risk, frequent pneumonia, and most commonly, the pain crises that are associated with impaired oxygenation of distal organs. As a result of current treatment, many patients will also have iron load, mostly in the heart, adrenal, and liver. There are 1 in 400 births of African American in the United States are with sickle cell disease, 1 in 36,000 in Hispanics, 1 in 123,000 in white. Our population, about 80% of our patients are African American, 20% are Hispanic. It is estimated that 100,000 patients in the US live with sickle cell disease. The median survival is 42 years in males, 48 years in females. Sickle cell disease kills worldwide about 500,000 people in the world. These are the areas of concentration of sickle cell disease in California. Let's focus here in this is Northern California, in Southern California, Antelope Valley, in Central and South Central area in Los Angeles, and another concentration is in the East communities here around Fontana, Manatee, these areas. This is the picture of survival of children with sickle cell disease nowadays. What you see here is that in Africa, 10%, in Nigeria, 100,000 children are born with sickle cell disease every year. 80% of them will not make it to the second year of life. In Jamaica, 84% will survival by 2 years. In the US, 94%. In the UK, 99%. So this change, the difference between what happens in Africa and Jamaica and the US is actually what happened due to the treatment. There has been a shift of the burden disease from the pediatric age to adolescent and young adult. And this is what we actually see in our practice. There is an increasing number of adolescent and young adult. The cost of the healthcare system in 2007 was roughly, as we can see here in the bottom, $890 to $2,500, and summed up to about 460,000 in 2007. A big part of this work was done by our colleague Julie Wolfson, who worked here and published extensively on the disparity of care between African American living in the fringes of the centers of the urban centers, and especially patients with sickle cell disease. This is probably the most telling graph for the need for something that I can show. This is the life expectancy for the next 10 years in each age group. And you see that while this is where African-American females and males are without sickle cell disease, females always have a longer life expectancy than males, but this is really measured in and this is where females and males with sickle cell disease are. This is the drive to start to try to find a solution to this problem. What are standard treatment currently? Mostly supportive care, and prophylactic antibiotics when needed, pain medication when needed, blood transfusion for acute events, hydroxyurea. This is something that has been shown about 20 years ago. It increases the production of gamma hemoglobin, therefore the production of hemoglobin F, and patients with hemoglobin F suffer significantly less vaso-occlusive condition. There is still the fetal hemoglobin F maintains a good affinity to oxygen, and therefore patients do not suffer the consequences of the disease. There are newer agents directed towards increased affinity of oxygen and therefore preventing sickling. With all that said, none of this treatment is curative. So what does City of Hope has to do with sickle cell disease? So what we have here, City of Hope has developed over the year expertise in hematopoietic stem cell transplantation and more recently in gene therapy technologies. Currently, hematopoietic stem cell transplantation is the only non-curative option for patients with severe sickle cell disease. There are early steps in gene therapy, but this is still an evolving field. We are involved in it, as you will hear in a few minutes, but this is not yet a proven way when bone marrow transplantation, hematopoietic stem cell transplantation, has been shown to be curative. This is a summary of what is known to date. And we can see that the CTN study with Metron-related donor, most of the studies, initial studies were done in sibling at very young age. There the results shows a very high rate of success in cure actually of the problem. This is initial studies with alternative donor, CTN MAD, about there were about 76% overall survival, 76 disease-free survival, but significant load of acute GVHD and chronic GVHD. A follow-up study with continued because it showed a good survival, patients survived, but very low disease-free survival. So that study was actually aborted. The Johns Hopkins with haploidentical showed 50% rate of engraftment and long-term event-free survival. New York Medical Center, also with haplo, showed a high percentage of event-free survival, but long-term follow-up is missing. Where do we see ourselves? This is the big universe or the big iceberg of sickle cell disease. We do population sciences and molecular informatics. Dr. Richard Weeks in population sciences does a lot of work on that. We don't do a lot of standard care. We have done it until recently, but this is not where our expertise lies. Our expertise is in developing newer modalities aiming at curing of sickle cell disease. This is our experience today. We have so far done 31 transplants. It is, to the best of our knowledge, it is the largest number west of the Mississippi. It's higher than any other center in California, more than Oakland, which is a center dedicated to sickle cell disease, and more than children or UCSF, Stanford, and UCLA. We had we have about 12 new sickle cell disease referred at any given time. We have about 8 or 10 candidates on every of our meetings, and there are 3 clinical trials that we will talk about in a moment. This is a summary of what we have done. So far, you see here, these are related, about 60%. These are metron-related. We have one cord, and this number, the haplo, is increasing. That will be the topic of our discussion. This is the age distribution to date. And adolescent and young adult will take the majority of the cases. And this is our survival to date, about 90%. This is the pipeline that we have and we will discuss today. The first one will be presented by Dr. Pavlovska. It's a pre-transplant immunosuppressive therapy before admission to the transplant itself. This is first in human mixed camelism study, and then we'll talk about gene therapy. So, Dr. Pavlovska.
Excellent. I'd like to talk about, to present our study, pilot study of pre-transplant immunosuppressive therapy for haploidentical transplant of patients with sickle cell disease, and nothing to disclose. And I think when we think about it, the transplant is the cure for the sickle cell. Then why don't we do it? Why don't we take every single patient with the sickle cell and just do transplant? There is very, very complex and complicated issues. And one of it is that the clinical course of disease is very variable. It means that the patient, we could not predict based on the family history of genetics composition, who is going to develop symptoms. Some patients can start having symptoms within first year of age and it progresses very, very fast. And some don't. It means that we could not take everyone to transplant because there are complications and take them for transplant with all the risks. And also, when we decide to go for transplant, very often those patients are already having a lot of symptoms. They are not the good transplant candidates. They already have pulmonary hypertension, they have strokes, they have cardiac dysfunction. It means that those transplants are very challenging and also can be life-threatening for the patients. And there is also high concern about transplant-related mortality. You know, like you have sickle cell disease, it is not cancer, and you are going to give patient chemotherapy, and there is risk of secondary malignancy, and we are putting the patients who can have even life expectancy is 40 years, and that can be pretty miserable. Still, the patient can die through transplant. It means that, you know, it's like the patient needs to be aware of all the risks coming. And also, there is difficulty in identifying a suitable donor. When we look within the registry, there is a lot of, we have difficulty finding the sibling donor with the population, and also it's very limited, about 20% of the patient have the unrelated donor within the NNDP. It means that first we need to find a donor. And I think this is which Hopkins tried to do, and you know we all know about haploidentical transplant developed by John Hopkins, and they are the first who start using the post-transplant cyclophosphamide, and of course it started with leukemia, and then they apply it to every single known disease in the earth. But between 2006 and 2011, they had 17 patients which had very long follow-up over 2 years, and 30 years of age, it means that 15 to 46 years. This is reduced intensity transplant. This is the original transplant with a little bit of like 200 of 200 of TBI and reduced intensity. No GVHD, only one patient developed skin, only acute GVHD, no mortality, 43% graft failure. It means that the patients are going through chemotherapy, the whole process, and 43% of those patients, they do not achieve the cure of the disease. And all patients have early pain post-transplant, because, you know, this is like pain. It's the issue when we deal with the sickle cell patients. For a year, they had issues with the pain and later on resolved. It means that we decided that we would like to look at the haploidentical option, and overall objective of our study is to expand the donor pool for sickle cell disease patients using haploidentical donor. The next thing is to cut down on toxicity, because those patients are coming with a lot of morbidity already. It means that we try to develop a reduced toxicity, however, myeloablative regimen, and achieve a consistent donor chimerism utilizing pre-transplant immunosuppressive therapy. I think in the Hopkins study, almost everyone engrafted initially, and only one patient who had the graft failure was the primary graft failure. All other patients were secondary graft failures. And what we have the pilot open study to evaluate the safety and feasibility of 2 cycles of pre-transplant immunosuppressive therapy, followed by myeloablative conditioning regimen and allogeneic hematopoietic stem cell transplantation from haploidentical donor. And once again, our hypothesis is that this regimen will provide successful hematopoietic and immunologic reconstitution, decreased acute GVHD and chronic GVHD. The acute and chronic GVHD is critical. Those patients do not need to develop GVHD because it's like trading one disease for another. You're coming with the sickle cell and up for the GVHD later on, and then overall improve outcome. And before we started the study, we already transplanted 4 patients, which we published, and we have 1 additional patient who also received the same conditioning regimen before we started enrolling on the study, just to see how feasible it was. 4 patients, it was just published this year. 4 patients received T-cell repleted haploidentical stem cell infusion. All patients engrafted with 99.9 to 100% donor chimerism, and they all have the stable engraftment for at least 1.5 to 2 years post-transplant, which it's really important. It's not going away, it's not fading. Time to neutrophil engraftment, 14 to 26 days, which is excellent. And I think for the haploidentical transplant, one of the pretty big challenge is the anti-HLA antibody, and this is very well documented with the patients who have this type of donor-specific antibody. They have the very high risk of graft failure. At the City of Hope, we have the protocol how to deal with it. 2 of our patients, they have high titer of the anti-HLA antibody. They received our management, and they engrafted day plus 16 at day plus 26 without problems, which I think we're pretty proud of the achievement here. And this is just from some few information why we are excited about the protocol. This is from the publications. We have 4 patients between 12 and 24 years of age, and we use bone marrow and peripheral stem cells, and the GVHD was really minimal. They have grade 1 acute GVHD and some very, very limited chronic GVHD of everything resolved, and all the patients are off immunosuppressive therapy. And the follow-up, it's between 1.5 to 2 years right now, and engraftment is excellent, 100%, and the last chimerism, it stays very, very stable. The one interesting thing is that those patients, they are haploidentical. Very often, if it comes from the father or the mother, the parents do have a trait, or the sibling very often have the trait. It means that the sickle cell percentage is exactly the same as the sickle cell of the donor. So they are not 0 because they have, but they do not have any symptoms, and pretty much they behave like the sickle cell trait at the time, and this is the percentage here. And just the quick schema. We start about 2 months before transplant. They have 5 days of Fludex. Then this pre-transplant immunosuppressive therapy, it's to ensure the stable engraftment after transplant. And then they have the second course, the same. And then they get the ATG, fludarabine, and busulfan. They get infusion, and they get cyclophosphamide day 3 and 4. Eligibility, and they need to have sickle cell disease for the clinical trials, they do need to have the symptoms, and ages 1 to 30 years of age, it goes up, and they are not allowed to have matched sibling or match unrelated donor, and this is pretty much standard what they are coming with neurological events, pain symptoms, osteonecrosis. And key points of the study, that they get 2 cycles of PTIS. There is the myeloablative regimen for patients with relatively good performance status, younger, less comorbidities, and they are getting post-transplant cyclophosphamide. Right now, we have 4 patients who are enrolled in the study and receiving treatment are doing very well.
So that is the first study. The second one is a trial attempting to achieve mixed chimerism through haploidentical transplant. So why mixed chimerism? This is the study. It is supported by CERM. It is also supported by the WANAC fund. Why mixed chimerism? Because it turns out that a patient with sickle cell disease who underwent transplant, even if they have a mixed chimerism with 80% recipient and down to 20% or even lower of donor cells, the sickle cell symptoms are completely resolved. It is not, there are a lot of theories why. Probably the major reason is that the red cell, the healthy red cells, because of their longer survivability, will outlast the sickle cell disease that the patient continues to produce. Based on that notion, we developed this program. It is based on work that was done, continues to be done by Dr. Defu Zheng in the Department of Immunology and Metabolism. And he has shown in multiple mice models that he can achieve mixed chimerism based on the method that I will describe in a moment. And this mixed chimerism is surviving over time. This is a list of the publication. I will not go over all of them, but it's a list of publications that they've shown that clinical symptoms of sickle cell disease were resolved when a person achieved mixed chimerism. This is the hypothesis. We can achieve immune tolerance through stable mixed chimerism, which will prevent GVHD and graft rejection. It has significant importance in areas that are out of the sickle cell disease, that I'll say only in parentheses, because if it is true, it could be used in organ transplant because you could achieve immune tolerance between the donated organ and the recipient if this point can be proven. This is from Dr. Defu Zheng. This is when he used Cytoxan and pentostatin. You see here the 2 populations, and when he added ATG, he used, here you can see the 2 populations, and then 1 population will disappear. And here when he used added ATG, you can see the 2 populations living side by side in chimerism. This is the haploidentical process for this protocol. This is the first in human. We have 3 patients that consented to go on the trial, but we have not yet started treatment on any of them. The donor will consent. We will mobilize cells with G-CSF and plerixafor. A portion of these are the mobilization days. This is the plerixafor, 2 days of plerixafor. By regulation of the FDA, a unit will need to be collected for a backup, and these are the 2 collection days. The collected units will go to Dr. Cardoso, Angelo Cardoso here for manufacturing. The product will wait for release testing. Once the product will be released, the patient will continue with the treatment. This is the process of the manufacturing. I will not bother you with that. Here is a summary of the treatment itself. This is on its face a non-monoablative regimen. It consists of 21 days of oral Cytoxan at a relatively low dose, 200 milligrams, 4 days in the blue dots of pentostatin, IV pentostatin, and 5 days of ATG. With the exception of the ATG, all this is planned to be done in the outpatient setting in the Alpha Clinic. For the first few patients, we will probably admit them for the day of transfusion just to make sure that they don't have any unexpected side effects. So this is a single center study, phase 1. We plan to enroll 6 patients with sickle cell disease. As I mentioned, we have 3 patients that we already consented to the study. The design is 3 and 3. Patient 1 and 3 will be staggered a month apart for safety issues. If safe, with at least 1 patient achieving mixed chimerism, up to 3 additional patients may be treated. And patient will be followed for 2 years post-hematopoietic stem cell transplantation. Primary endpoint of the study is to evaluate the toxicity of this new regimen. The regimen by itself is based on some studies that were done on the NIH for other types of transplant where it has been shown to be safe, with the exception of the major element that we do, which I kind of overlooked on the manufacturing. In manufacturing, the most important part is that Dr. Zeng's lab showed that when he removed CD4 positive cells from the graft, it's not a total T cell depletion, it's a selective CD4 depletion. Engraftment occurred without GBHD, and the engraftment in animal models was lasting, so there was no leg rejection. Similar studies that are looking at mixed chimerism, inducing of mixed chimerism, have to use total body radiation because without it, the recipient will eventually reject. And we are aiming to see if that process will enable us to do the transplant without rejection and again, without GBHD. We want to see induction of mixed chimerism, at least 30%. 20% is what was reported in the literature. But in the literature, there are cases of 11% mixed chimerism. We have a patient who is down to 10 or so and has no signs of GBHD. We selected the 30% as a safety. And we want to see the feasibility of manufacturing the CD4 T-cell depleted study. One thing that I mentioned to say, the first study, the study presented by Dr. Pawlowska, although reduced toxicity, is a myeloablative. This study targets, generally speaking, older and sicker patients. So patients are eligible, there is an overlap between 18 and 30, they could go to either study, but if they have comorbidity, they will be directed to the mixed chimerism study. Patient above 30 will go to the mixed chimerism study. Our initial goal was to have patients even older than 45, but based on the recommendation or actually the direction of the FDA, they asked us to limit the age to 45 and go up the age if the results of the up to 45 will justify. Needless to say, if the study shows promise. There's no reason to limit the ages at either direction. Why should the younger patient go through a myeloablative regimen if a non-myeloablative regimen can do the same job? I think I already spoke about that. These are the secondary endpoint: kinetics, chimerism, sickle cell hemoglobin levels. We will monitor the sickle cell hemoglobin levels periodically. That leads us to the next topic, and that will be gene therapy, which will be presented by Dr. Cardoso.
I'm not sure the pointer works. Yeah. Okay. So I'm going to talk to you about gene therapy for sickle cell disease and the work that has been developed by our team and other collaborators at City of Hope. I have no conflict of interest to declare. And we go back to the initial slide from Dr. Rosenthal about the genomic defect on sickle cell disease. So as he mentioned, the sickle cell disease is a monogenic disorder, and the mutation is a single base pair mutation in exon 1 that leads to substitution from glutamic acid into valine, the beta sickle globin. And this mutation is sufficient to alter the physical properties of the hemoglobin tetramers that are formed with the two alpha globins associated with the two beta sickle globins. And the important fact is that this single base pair mutation drives the pathophysiology of sickle cell disease. So you can imagine that the easiest thing to do in the ideal world would be if we have genome editing to just go to the hematopoietic stem cells and do the editing of this defect and replace with the correct amino acid, the correct nucleotide, and then you fix the disease. So this will be the long-term goal. We are not there yet. There are strategies of genome editing that give hope that this one day will be possible. But today I'm going to focus on the ongoing efforts that are in the clinic or going into the clinic of gene therapy for sickle cell disease that are based on use of lentiviral vectors to do globin gene correction or globin gene addition on hematopoietic stem cells. And in these studies, the goal is to collect hematopoietic progenitor stem cells, C34 cells from patients with sickle cell disease, ex vivo modify these cells and then transplant them in the patients. So to understand the strategies that have been developed, it's important to talk a little bit about the biology of globin expression and regulation. So during fetal life, the prevalent form of globin is the globin gamma, and the fetal hemoglobin is a tetramer of two alpha chains and two gamma chains. But then after birth, there is a gamma to beta hemoglobin switching in which the fetal gamma globin expression is silenced, and then the beta globin becomes dominantly expressed, as you can see here in green. So adult hemoglobin, you have two alpha globins and two beta globins, and then in sickle cell disease, these beta globins are mutated. Now, it's important to understand that the regulation of globin expression is tightly controlled by genomic elements like this locus control region, which contains 5 HS sites, so for DNA's high supersensitivity sites. And these sites are gonna loop the DNA, and with a complex of transcription factors are gonna control the expression of the globin genes. So in fetal hemoglobin, they drive the expression of the gamma globin genes, and then in adult hemoglobin of the beta globin genes. And an important factor in all this is a zinc finger transcription factor called BC11A, which is a major regulator of the gamma to beta hemoglobin switching by silencing the gamma globin expression on human cells. And you are going to see that all this information and all this knowledge on the regulation of the globin expression genes are important for the design of the vectors that are being used now in the clinic. So the strategies that are being used are strategies for gene correction of gene addition and include strategies like using beta globin expression cassettes to correct the adult immunoglobulin expression. You have synthetic beta globin variants with anti-cyclic mutations like E22A, T87Qs, G16D. These increase the affinity to the beta globin subunits and make a more stable adult hemoglobin, providing an advantage compared with the sickle hemoglobin. Other approach is the transfer of gamma globin expression vectors to drive fetal hemoglobin expression. And one of the strategies I will show you is using an hybrid gene cassette with the gamma globulin coding regions and beta globulin non-coding regions to stabilize the vector. And the rationale for this approach is that the fetal hemoglobin, the alpha-2 gamma-2 tetramer, has more potent anti-cycling properties than the adult hemoglobin, and also have a higher affinity for the binding the oxygen. And it's known that patients with sickle cell disease that have high levels of fetal hemoglobin have attenuated disease. In fact, there are reports of patients with sickle cell disease that have a condition called hereditary persistence of fetal hemoglobin, in which the levels of fetal hemoglobin reach 30% or above. And these patients, despite the fact that they have the sickle cell disease mutation, they don't have any evidence or any clinical signs of sickle cell disease. And then the last approach is to use a vector which contains a hsRNA to specifically knock down BC11A. So the idea is if you silence the BC11A mediated repression of the gamma globulin gene, then these patients are going to start to produce higher levels of the gamma globulin and therefore of the fetal hemoglobin. So this is a cartoon of the 4 vectors that are presently in the clinic being tested in phase 1 trials. So the first vector is the vector from Bluebird Bio, and I will show you after a little bit about the clinical trials. So this is a vector that has the beta globulin gene with anti-cyclic mutation, the T87Q, here is the asterisk. And as you can see, all these vectors in common have the fact that they all contain HS sites from the locus control region to drive erythroid-specific expression of the globin genes, but also high levels expression of the transgene. Then another vector is this one here called BAS3 that was developed by Don Kohn at UCLA. So this vector has the beta globin genes with 3 anti-cyclic mutations. He also has an insulator here. And as I mentioned, is now in clinical trials. And this third is a different approach. In this vector, again, you can see the HS sites from the LCR. And this vector is a hybrid vector that has the gamma globulin gene, the coding regions, and he has the non-coding regions of the beta globin to stabilize the vector. It was developed by Poonam Malik and is now in clinical trials. And finally, this is the David Williams work. They use a shRNA to silence BC11A. This vector also has LCR motifs and is also now in clinical trials. So now to summarize the trials that are ongoing. So let's start with the with the lentiglobin BB305 from Bluebird Bio. They have 2 clinical trials. This one was using bone marrow hematopoietic stem progenitor cells for sickle cell disease. This trial is still active but non-recruiting. And the strategy now has changed to the trial above, in which is open for both beta thalassemia and sickle cell disease. Initially was using bone marrow hematopoietic stem progenitor cells, but right now, even in sickle cell disease, they moved to plerixafor mobilized hematopoietic progenitor cells collected by apheresis. The goal is to enroll 29 patients. This trial is active, and the condition regimen is myeloablation using busulfan. So another trial being conducted by Poonam Malik at Cincinnati Children's Hospital is using the hybrid gamma globin beta globin vector. They use reduced intensity with melphalan for conditioning, and the target cells are bone marrow CD34 cells mobilized and modified ex vivo for then transplantation. Then this trial is DONCON trial at UCLA. The goal is to enroll 6 patients. It's an active trial and uses the lentiviral vector, the beta globin with the 3 anti-cycling mutations, also bone marrow hematopoietic stem progenitor cells, and is also a myeloablative regimen with busulfan. And finally, the Boston Children's Hospital trial. The goal is to enroll 7 patients. It's active. They treat until now 3 patients. And use the lentiviral vector with the BCL11A shRNA, myeloablative busulfan. This is wrong in the printouts, but this is the right regimen that they use. So, but of course, there are significant challenges in developing gene therapy for sickle cell disease. And these come from understanding the disease, and we are also learning from the results of the first patients being treated in the trials that I show you. So one of the challenges is that these patients, they go through repeated bone marrow and bone crises, a vascular necrosis, and these affect the number of CD34 cells in the bone marrow. So there is a reduced number of cells that are going to be available for bone marrow harvests, and can potentially have an impact also of deficiency of mobilization of these cells for apheresis and then for gene modification. The question is the fitness of these cells for gene modification. So these cells live in an environment, and this point here, in chronic inflammation, and these can potentially impact on the quality of the stem cells from these patients, but also a potential impact on mobilization, and can greatly affect engraftment. Now, another important point to consider is that in beta globinopathies like sickle cell disease or beta thalassemia, the gene-modified hematopoietic stem cells do not have a survival advantage. And this is very different from what you have in monogenic diseases of the immune system, like immunodeficiencies. Like in SCID, for example, gene-modified hematopoietic cells, when you transplant them, they have a selective advantage. So you can have a smaller number of cells, you can have a lower levels of engraftment, and have a successful gene therapy, but these don't happen in these diseases. So what's happened is you need a higher degree of chimerism of the gene-modified HSPCs, and usually means you have to start with a higher dose, and you mean that you need to have cells that have a high quality and they are fit to be gene-modified. So this is another challenge. So other way to try to work on this angle is you have to improve strategies to transduce these cells, and you need to have a higher level of globin expression per vector copy. So all these are areas that are being worked on to try to make better vectors and improve the efficiency of transduction. And finally, we need to have, it's very clear, you need myeloablative conditioning to transplant for the engraftment of sufficient number of these cells, and these you are gonna have damage to a bone marrow that is already damaged by the sickle cell disease. So these are some of the challenges, and now I'm gonna move to show you some of the work that is being conducted at City of Hope to address on some of these areas and try to learn more how can we move our gene therapy for sickle cell disease project forward by trying to look at different levels. So I'm going to talk about two of them. We don't have time to talk about our program of genome editing for sickle cell disease, but let's start by the PISMO trial. That is a trial looking at mobilization of autologous sickle cell disease patient hematopoietic stem progenitor cells using Plerixafor. So this is a trial that we hope to open by the end of this month. And the goal of this trial is to do a systematic analysis of the mobilization, the kinetics of mobilization of C34 cells from sickle cell disease patients using Plerixafor. So the new thing, so the goal is that the patient will have a dose of Plerixafor at time 0, and then there will be blood collections at different time points, and then after 24 hours, a second dose of Plerixafor also with blood collections, and this will allow us to study the kinetics of mobilization of CD34 cells in sickle cell disease, and we can learn whether it's altered or modified compared with what we know from normal healthy donors. So the other novelty of this study is that until now has never been tested whether you can use 2 sequential doses of Plerixafor 24 hours apart. And this is important because looking at the very limited available data and looking at our manufacturing data and what is known about engraftment, we believe that we will need an approach like that to have sufficient number of CD34 cells to do gene modification and then to achieve sufficient number of engrafted gene modified hematopoietic cells for these patients. Now, one thing we are going to do here, very important for the study, is at a 6-hour time point. I think now it's going to be 4. We just changed this last week. We are going to have a larger collection of blood, and we are going to purify C34 cells from this blood from the sickle cell disease patients, and we are going to do studies to look at the fitness of the cells. We are going to look at the phenotype of the cells, subsets of stem cells. We are going to transduce them using a lentiviral vector. This is a new lentiviral vector that has both gamma globin cassette, but also as a shRNA that we're going to use to allow the selection of the gene-modified cells using 6TG, 6-thioguanine. So the goal of the study then is twofold, is to learn about the kinetics of HSPC mobilization in sickle cell disease patients and study the fitness of the cells for gene modification and for selection of the gene-modified cells. So the other study that I'm going to share with you is being developed by Nadia Carlesso group at BRI, and the goal is to look at the bone marrow niche in sickle cell disease. And this study is possible because of the studies being conducted by Dr. Rosenthal and Pawlowska with the clinical trials. These allow the opportunity to get specimens, bone marrow specimens from these patients before transplant and after transplant. We also can look at donors, including donors with sickle cell trait, and also Nadia is looking at the sickle cell disease Berkeley mouse model. So the goal of this study is to analyze the bone marrow niche for cellular components, inflammation markers, cytokines, the function of the stem cells, and the goal is really to characterize and quantify the degree of inflammation in sickle cell disease. This is very preliminary data, but just to show you, we developed a multiparametric flow panel in the lab that allow us to look at 14 or 15 different parameters, and that allow us to look at different subsets within CD34 positive cells. We can distinguish, for example, the cells that are long-term HSCs from MMPs and other subsets. And this is very early because the number of patients is still very limited, but we see some differences between sickle cell disease and bone marrow from healthy donors, and some populations of cells like the CMP, EMP, GMP seem to be really very rare in these patients compared with normal donors. And this is just colony assays from comparing a healthy donor with a sickle cell disease patient in red, and in green is the same patient, but now 2 weeks after transplant. As expected, we have a big jump in the expression in the CFU colonies as expected. But the early data suggests two things: that in fact there are quantitative differences in the bone marrow, in the stem cell compartments between sickle cell disease and normal donors. And now there's also some preliminary data showing that there are changes in cytokine profiles, and there are activation of inflammatory pathways in sickle cell disease stroma with activation of NF-κB and Mir155. And, and then just the goal of this study is that if we learn more about the niche in the sickle cell disease, the goal is to see whether there will be therapeutic approaches that we can use to protect the stem cells from inflammatory damage, and that can be used to improve long-term engraftment of the gene-modified cells. And I finish with this slide that is, we believe that the beta globinopathies, sickle cell disease, and beta thalassemia are the most frequent monogenic diseases that can potentially be cured by gene therapy interventions. And I think this is a big focus of research of our and many other centers in the next few years. Thank you.
We have one more moment. I just want to say that the work that was presented today is only part of the work that we are doing in sickle cell disease. We didn't talk about the big effort that, one of the issues that we face in our patients with sickle cell disease is that there are a lot of psychosocial issues that patients with sickle cell disease bring with them at the time of diagnosis. The low self-esteem, the mentality of sickle cell disease. So we have an effort together with our colleagues in supportive care to develop a well-systematic way to deal with sickle cell disease on multiple of areas. And then finally, there are multiple disciplinaries at the City of Hope that are involved in this project. Dr. Defoe and Dr. Riggs on the Department of Metabolism, gene therapy, Dr. Zaya and Dr. Cardoso, Dr. Carleso, which her work was done here. Our colleagues in hematology, Kaiser Hematology, who are our major source of referral. Our colleagues in pathology and transfusion medicine, who are an essential part of it. And I want to take this opportunity to thank everyone for their help. Thank you.